So thanks for inviting me and giving me the opportunity to talk about my research. And the name of my talk indeed is because think about precision medicine. If you ask students, people will say, well, you have a marker, that predict mostly cancer, something that is clearly associated to genotype. And things are changing. But what we do is slightly different. And what I want to talk today, not different in terms of what we use, but the data sets are different or it wasn't done yet. So I thought to talk about two things. One is machine learning in malaria, and the other one is tools of a database of immune-mediated inflammatory diseases. It's important to say, and I think it's a wide field, and I think it's really thriving, and no one does something alone. So the malaria work was actually, I would mostly say, single-handedly done by Collins, who is a perfect PhD student at WACBIP, it's the University of Ghana. And the IMED Bio work is part of a very large MRC consortium, but there's basically the data team. So it's all, I think, it's important to state out that in our days, these are mostly, if you work with data, with clinical data, that is mostly it's our big consortium. So let me just start the time because I don't see the time. And I said to Adam, I'm normally quite good on time. Let's see if I am right with that. So let's dump it. I think most of you obviously, hopefully will know about the issues of malaria. Still 400,000 deaths per year. I mean, COVID has more this year, but overall this is every year and there's no real solution. Maybe now a vaccine, but there are other problems around it. There are 3.2 billion people at risk and 200 million infections every year. And we have drugs. And the problem is, it's very easy to diagnose severe malaria. It's easy. The clinician told me that. You're basically dying, and then it's very likely to be malaria. People get treated. But there's a difference between uncomplicated malaria and non-malaria diseases. And these basically have very, very similar symptoms. So our aim is, we're using existing databases or data sets or Excel spreadsheets actually. Can we kind of determine the classifier to say, hey, this is severe malaria, uncomplicated malaria, or non-malarial diseases? And obviously, this would be something very nice because this is an issue for the clinicians to determine the differences, especially around uncomplicated malaria and non-malarial infections. Okay, so we thought, with Collins, why we don't have a look at that? And we took a data set from Ghana, 3 different positions, and actually quite big. And he basically then did different comparisons. And overall, we have quite good sample sizes. Obviously, if you think about normal machine learning, you would always, the more the better. But these are clinical studies, so we were quite happy with the data we have. And we just, let's go. But I think very important is something I really have to emphasize, and it's very important, is also especially for the second project I talk later, is what is actually data curation? How much does different type of steps taking? So obviously the first thing is you have to have data acquisition. So this was outside of our study. Now we need to have, or what actually Collins did, he needed to pre-process the data. He needed to harmonize them, to check them. We then saw actually that we had far more samples, but the samples were done on different machines. So some of the parameter didn't fit because we're done with different machines. And it was not just a batch effect. It was a real difference that let us discard the data. Obviously then, once you have that, you can just do your machine learning. We used here 6 different approaches. We run on the data, and then normally you try to understand the model, the black box, what actually does the machine learning do? But in terms of time, actually, we try to put estimates on it. And obviously, it's never really exact. But most of the time, obviously, it's a sample preparation where people go into the field. But this is done by many people. This here, the 20%, is done by one person to really kind of curate and harmonize the data. And the better this is, the less you lose data, the more complete the data set is, the more you can, the better actually your predict is going to be. And it's just something I thought to highlight because it's important for many precision medicine projects we do. So here are the data, and you can see we have the different parts of the non-molecular infections and overall the sample size. The data overall were quite nicely balanced, apart of location and age. And there's something I'm going to come back, obviously, to see if we have every confounder there. So Collins, and in a clinical manifestation on the non-malarial infection, we have a little bit a mix of everything. Nothing really stood out. So it's a good mix in terms of what's there. So we didn't think there was any bias that could have interfered with our predictions. So Collins set then up like an artificial neural network here, 3 layers. He trained it, as said. He did model interpretation with Lyme. He did a lot of testing. And then actually, after we submitted the paper, we needed to do a lot of optimization that he did. And you could see how well actually the model converges. So overall, using existing tools, and this is obviously all available, the code that he used. And I shouldn't take any credit here because he did a great job. And what are the results, actually? So overall, our prediction works quite well. So you see here our neural network. The severe malaria with the non-malarial infections, the training is actually extremely good, nearly 1, and the testing is quite nice. And here we see we have a bit, but overall around 0.8 was actually a good predictor. So it would really help the clinicians if we could use that as a support in clinics. The other one are similar-ish, but not as good. And the support vector machine was actually also quite nice in terms of the comparison of the uncomplicated malaria, the one actually with more difficult for the clinician to distinguish. So if you do a ROC analysis, you can see how it behaves overall quite nicely. Could obviously be better, but with the parameter we had, this is what it is. Where else the severe malaria versus non-malaria infections is actually pretty much binary, which is kind of obvious if you think about it. When people come in with all the symptoms of malaria versus a kind of more milder infection. The question now was, but what actually does our model classifies? And so we decomposed basically, and you could see that the classifier for uncomplicated malaria was driven mostly by platelet counts and blood cell counts. Or here you see the other one, the neutrophils percentage. So these were the variables that drove most of the model, but all of them actually count in quite nicely. And the same actually come up even in the severe malaria, even more. You see the red blood counts, etc. So overall, we were able to kind of find the parameter that actually drive our neural network. So as I said before, one issue obviously was that our data set is not biased. So as I said, there were published data sets before that were done for other studies. We just thought, why not reanalyzing? So they weren't really kind of constipated to do this kind of work. So what we did actually, we subsampled from. So Collins basically said, let's have a look to get similar distributions. And you could see that overall the accuracy doesn't really drop. And most important, I was really worried about the age, because obviously younger children get more severe malaria, more likely. So there's a huge age bias. And but you could see once you normalize it down, the accuracy of our models is still actually quite high. So overall, we were quite happy with the results. Actually, we're the first, as far as we know, and that basically used clinical malaria data on hematologic parameters to kind of classify. And then obviously you're going to ask, well, is it really feasible? How likely is it that you could go in Africa and take the blood and do things? I think it's going to come, right? People will do blood tests. We can, especially if it's uncomplicated malaria, we can wait a day or two. We can really have the blood test. Think about travelers coming back from Africa that have mild symptoms. Do you want to just treat them and generate more resistance? So overall, we think that we will have an impact there. And if you're interested in the paper, just have a look. It's published in BMC Medicine. And just acknowledging this part, obviously, as I said, Collins did all the work, and this was a collaboration together with Gordon, Lucas, and Saiku, all from VACPIP and the different funders. So now changing slightly gear and going into more diseases that are more existing in our spheres, basically immune-mediated inflammatory diseases. And you will see there's similarity between the project with malaria, because basically we already did what we want to do here, but this is slightly bigger. What is the problem with chronic diseases? It's basically what you have is that the immune system attacks yourself. So the immune system kind of somehow decides, I'm going to attack myself, or attack the body that hosts me. And there are different diseases like rheumatoid arthritis, and mostly the joint, psoriasis is the skin, etc. And there are several ones. And it's increasing, it's 2 to 5%. And what makes it more difficult to work with compared to other diseases, there's just around 30% of inheritance in it. So basically, there is obviously 30% SNP-based wise, but the rest is actually other factors like maybe weather, maybe smoking, but we don't know that. But this makes it actually more difficult to apply just predictors because things are just more complex. So to kind of overcome that and try to put things together, there was this MRC project that was led by Ian McInnes, a colleague here at the university, and he said, why don't we bring all the IMID cohorts together under one roof, basically, and in the UK, build a big database and try to harmonize the data. This got funded, the funder liked it, and I arrived slightly later into the project, as explained later, to help with the data integration. But the focus basically, how can we cross-learn between diseases? What is similar? Are there specific endotypes, something that is similar between the data sets? It would be good to generate standards and best practice. How would you define a clinical study to capture most of the data? How is actually the best way to store these with database? What kind of tool can you do? Can we borrow some tools of other projects? And then there's the outcome. We'll have around 30,000 patient data, predict disease of onset, obviously using machine learning and AI into integration of multi-omics. This is an ongoing project. It's basically we an open consortium, so people who want to collaborate are invited to contact us and propose some projects. And the biggest problem we have is actually that we needed to create the data, because what we have is it's a lot of different cohorts that are coming together with different technologies. You have microarray, you have RNA-seq, you have some marker genes here and there. Obviously, the data weren't harmonized. It means different, like for example, smoking is captured differently. So it's a lot of little nitty-gritty things you have to solve and get over. Then the question was how we can sort. So what we did, we basically integrate everything in our Transmark database, which will come in a second. And the idea is to have it all in a data warehouse. And from there, people then can actually directly assess the Transmark, something I'm going to show in a second. Or they can basically do some Shiny apps to do some analysis. The Transmark, the method we use, is we didn't invent it. It's an open source project that exists. It was developed by G&B Johnson. And it's basically based on different projects. So you can, if you're more interested in that. And I wasn't involved in the decision to use this method, but it does what it does. It's some good sides, some bad sides. What important is it's open source and being developed. Let me just get some water if I find it. No, I don't. No worries. So important, as I said, is all the point. How can you then curate it? And remember when I showed in the malaria is how can we kind of not harmonize this data? And the malaria was one PhD student who probably drank a lot of coffees for some weeks and did it. Here it was a bit more of work. So Kevin Ross, who worked with me, he basically for 2 years, he harmonized a lot of data. And you must imagine it's not like the people have computer and have dropped down ways to or normalize every cohort in every different part of the hospitals around Scotland, in this case, basically wrote down by hand things that were then put into an Excel sheet. So you can imagine how things can go wrong. And together then, with the normalization, together with Mike Barnes' team, we basically, for the last 2 to 3 years, normalized the data. Now we're on a point where we're actually quite happy with 9 data sets in the Transmark that we can analyze. So what can you do actually in Transmark? And this is now what I'm going to show is mostly for non-bioinformatician, I guess, like clinicians who want to just play around with the data. They don't want to just go through an Excel sheet. They want to ask maybe, I don't want to say stupid question, but basic questions. So what you have, if you open Transmark, you have here, for example, I hope that you see my mouse. You can see the SERA data set, and you can digital data, and you have the different time points that were captured over the different years. So, and you have different comparisons, you have some advanced R, etc. What you can now drag and drop different comparators into these fields. So you can see here, for example, you can have a comparison. How is the difference between males and females? So you just drag the female, in this case, the males here, and actually, what do you want to compare? So for example, you can compare a DAS score. So DAS score is is a disease score that is a calculation of how tender and swollen your joints are, so 28, and with a kind of general idea of inflammation. So depending on that, I think it ranged between 2 to 10 or 2 to 11. You can basically now add this up and you put it in Transmark, you press go, and it shows you actually the differences. So it shows the difference in how many samples you have, and then you can start analyzing the data, which is actually quite basic. But on the other side, if you think about it, often these data are not accessible in that way. You can then actually look at the profile of the DAS score, for example, here, and you can see most of them are 5, and normally remission is just around 2.6. So this is basically means that you're okay. So most of the patient in baseline actually were obviously sick and had rheumatoid arthritis in this case. But you can obviously some more. So for example, here we compare the DAS, the expression of specific protein in whole blood, and you can actually just drag and drop it, and you can see the difference between female and male, and you can see if there's a difference in specific parameters. You can do different subtypes. So overall, you can play around with the data reasonably easy. And what we did 3 or 4 datathon hackathons, where we basically had clinicians coming in and we showed them how to work with the data. We give them accounts and they can work on the data set. We also did an industry workshop where we worked with our partner and gave them access to the data. And overall, the feedback was actually quite positive. But then obviously you think, well, that's not very bioinformatic-y. Is there not a more automated way to look at the data? And obviously you could just dump the data and put it into R, or you can build an R Shiny app. So the R Shiny app here was done by Raphael, and what it does is basically sits on the Transmark, and which is nice because it's not just for our data set, it's for all the data sets you can use. And he played a little bit around, so you can go through the different points and look at different data sets. And what we did actually, let's try to make an analysis that is meaningful. So what we did is we're exploring interferon scores across the different IMEDs. So what we try to do is we try to combine 6 or 5 different data sets. So we have an universal disease activity. We have that from the data from the age. And what we also have, we calculate interferon alpha and interferon beta score. And this is basically a summary of specific genes. So if you put specific genes together, you can kind of simulate the interferon alpha and interferon beta score. This was actually done, if you're interested, that was proposed in this publication here. So what do we get out? So first, we see different diseases. So we loaded in, this is a lupus data set directed for EBI. So basically, we download the data, imported it into Transmark. When I say we, it's the royal we, it was Sandra. And then now we have different IMEDs, so rheumatoid arthritis, Sjögren's, psoriasis, and also lupus. So it's clustered. So it's base clustered on disease activity on top, interferon beta score, interferon alpha score, and the age. And then you think, hey, this is super basic, right? But the interesting thing is we do see different clusters. So we see 10 different clusters. So you can see here the distribution on the violin plot about the age, disease activity, the alpha, interferon beta score. If you go through that, what you can see is, for example, here, the cluster 5 and 6. First, obviously, we have far more for the lupus data, but we see mostly younger people. So here you see the younger people, but they have a higher disease score. But why does it fall into 2 clusters? And one cluster has a similar alpha, the interferon alpha, but one cluster has a different. So it seems to be higher. So this difference actually split the 2 clusters. So why is that? We don't have to answer that. It's mostly an observation. What are the other clusters? Here you can see cluster that's mostly done by lupus. And then Mike went back and had a look actually at this patient. These patients were actually mostly treated. So it could be that these actually had lupus, or due to treatment, they looked similar in terms of their score than the lupus people. A very small cluster, but it's very nicely mixed with the different diseases here. And this actually, as you can see here, it's a good mix. It's small, but a good mix of age. And you have a high alpha, interferon alpha and interferon beta. So now we could actually go back and see, are these people maybe that have high inflammation, or whether drugs didn't work. So overall, with a simple comparison, we're actually able to kind of really dissect the data into 10 different classes. And we discussed a lot with Mike what this actually means. And he postulated this actually could that be pan-IMID endotypes? So could it be clusters where you can cluster the patient into 10 different classes? So you can cluster them depending on age, depending on disease activity, and then the interferon scores. But we wonder, why would you want to do that? What is the help? Well, this is actually all about precision medicine. Because assuming on the left-hand side, you have basically the different, you have the total of your patients, right? And what you have different endotypes. So specific subtypes of people with specific diseases. And if you don't know the different endotypes, you just treat specific amount of people, right? Here. But obviously, as the endotypes are different, the people don't respond the same way. The responders are limited to the correct endotype. If we know the endotype and we can predict it and help the clinicians, we obviously would treat the right group of patient and will A, treat more relevant, and B, we would also have more success. So this is the line of the work we're doing with IMID. Obviously, so in conclusion, we built actually a huge data warehouse. We curated so far 9 clinical data sets. We still welcoming more clinician into it. So hopefully this will grow. We are close to finish our first funding cycle and hope to get more funding. We develop methods to kind of analyze the data, our Shiny apps. We have protocols. So hopefully in future, if people do the similar work, we're going to share the best ontology. And actually what we're doing at the moment is we decompose complex diseases into the different endotypes to help actually people, or to think about, could we help clinicians to kind of stratify patients to treat better? And then actually we're coming back from off the beaten track to actually something we understand far better, where we actually have specific groups to target and really focus on specific people. So with that, I would also like to thank the BITS collaboration here. There are 50, 60 people work. So here, the main PIs and obviously Mike's team, who did almost of the work. And with that, I'm actually finished. Thanks you very much for listening. And also here, the people from my lab. Thank you.
Thank you so much, Thomas. It's refreshing to see an application of precision medicine to other fields than cancer, because cancer is the obvious one. That's what everybody talks about. But here, it turns out that actually we can also do these things in other fields, such as autoimmune disease and malaria. My specific question was going to be about what we've learned from working in the cancer field. We know that it's much easier to discover a method that will be useful to stratify patients than actually to put it in practice. And the reason seems here is that as soon as you start working with patients, things become really complicated. You have to have collaborations with clinicians. In cancer, these would be surgeons, but I imagine in malaria it would be more kind of tropical disease experts or GPs. What do you think? What do you think the precision medicine in malaria, precision medicine in autoimmune disease can learn from what we've seen so far in the cancer field, and when is it going to be practical for somebody coming back from sub-Saharan Africa to get tested for severity of malaria even before they develop symptoms?
I think the malaria is very early day, because obviously what you need more data to be able to do, and obviously there's far more interest in our society and the industry to go after cancer than to treat malaria. This is why I'm sorry, I don't want to be cynical, but I guess malaria is probably similar difficult to treat than COVID, but COVID we seem to have under control. And in malaria, though it's a parasite who evolved and shaped our genome heavily, I would love to argue that if we would put the same effort into, we could get the same effect. So I think there is a question of interest. I think so. In terms of learning, I think we just have to see what happens. And but obviously, as I said before, the main track is mostly going on to finding SNPs, finding markers. So, and if you can find markers, then you go into the field. But we actually went the other way. We started with big cohorts, and from the big cohorts with clinicians, we try to deduce what are actually endotypes. So what do we have in similarity? But what do the patients that have the similar endotypes have in common? How can we target that? And and I mean, cancer is also the treatment is different, right? You need
yeah, it's much more varied as opposed to malaria.
Yeah, let's maybe IMID diseases. I think it's more tricky. I think in malaria it's quite straight. You use the drugs and you treat people. That's fine. And the worst case, if you're not sure what to classify, you just treat. It's better to treat malaria than not to treat. But in IMIDs, it's more difficult because the drugs don't work. So what we try to do is to basically go into to understand which drug works for which person. And you can't, it's not a surgery like in cancer. You can't say, oh, you have to operate now, or your risk is 85%. So what you do here, it's the own immune system that attacks you. So I think it's obviously there is similarity, but in my view, as a data scientist, the tools you have afterwards are slightly different. And as I said, it's obviously there are the clinical data, but if you think in terms of inheritance between the way you find your markers in cancer is far more straightforward than you would find it with the IMIDs.
Yeah, no, that's really interesting. Thanks for this perspective, Thomas. It's definitely good to understand the differences, because these fields, these indications are indeed very different. We've got a question from our mutual friend Vicky from Glasgow, which is really just about data curation. So Vicky's asking, given that data curation clearly is the big bottleneck in scaling up a data bank, and you know in your previous question you said, oh, we need more data in order to actually make any meaningful progress. So how can you scale this up? That's Vicky's question.
I think that's a very good question, Vicky. I think the issue is that people didn't compile the studies thinking of precision medicine, and I think the way forward, and it should actually be possible to kind of have an ontology. So something we're going to propose as one of the outcome. What is an ontology to capture clinical data from IMIDs? And then rather than people having to write it down and then it's typed, have a form, get people. And I think the question, is it feasible to have hospitals with computers where people, or maybe when like a tablet, where people actually, when they capture the data, it goes directly into the database. And then it says, hey, it can't be that the DAS score is 20 because the maximum limit is 11. So if you put a little bit more intelligence into the system, in my view, to capture, then actually you would automatically have automatic curation. We looked into some machine learning to how we automatically improve. We obviously did some of that, but it is depending how noisy it is. And the thing is, every cohort is cross-hospital, and some hospitals do it that way, some hospitals do it this way. So I think it's just the nature of the beast that it will be mostly, there will be a huge part, there will be always a huge part of manual. So therefore, it wouldn't scale. I mean, you could think about outsourcing and get community annotation or curation in, but then you need a specific of knowledge of what is possible. So I think down the line we'll get better. And just with optimization and having more computers in NHS system, at least for the UK, things will go better and more automated, which then obviously will help.
Thomas, thank you so much for this answer and for the talk. In fact, it was very refreshing to see something unexpected, something off the beaten path.